Hello friends, today I would be teaching you all about biotechnology principle and process. So let's get started with the video. Biotechnology deals with the techniques of using live organisms or enzymes from organism to produce products and process useful to humans. You have to learn the definition. You should learn the definition of biotechnology. Okay. European Federation of Biotechnology has given a definition of biotechnology that encompasses both traditional view and a modern molecular biotechnology. The techniques of genetic engineering include creation of recombinant DNA molecule. What's recombinant DNA molecule? By use of gene cloning and gene transfer. Gene cloning and gene transfer. Making multiple identical copies of any template DNA is called as cloning. Multiple same copy like twins, these are called, the process is called cloning, and the progeny that is being produced by this process of cloning are called clones. The autonomously replicating circular extrachromosomal DNA in a bacteria is termed as plasmid. Definition of plasmid: replicating circular extrachromosomal DNA called as plasmid. The first recombinant DNA was made by linking gene encoding antibiotic resistance with native plasmids of Salmonella typhi. Firstly, it was done with Salmonella typhi. Stanley Cohen and Herbert Boyer made first recombinant DNA molecule in 1972. Who made? Stanley Cohen and Herbert Boyer. Molecular seizures in recombinant DNA technology are the enzymes restriction enzymes or restriction endonucleases or restriction exonucleases. Mainly endonucleases are used. DNA ligase enzyme acts on digested DNA molecules and joins their ends. DNA ligase acts as a glue. The two enzymes responsible for restricting the growth of bacteriophage in E. coli are restriction endonuclease and methylase enzyme. What? Methylase and restriction endonuclease. The first restriction endonuclease discovered was HIND2. HIND2 was the first restriction endonuclease. First restriction endonuclease was HIND2. Today, more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. How many? 900 restriction 900 restriction enzymes from 230 strains of bacteria. Restriction enzymes belong to a larger class of enzymes called as nucleases. There are two kinds: exonucleases and endonucleases. Exonucleases enzymes remove a nucleotide from the ends of DNA. What? Exonucleases works on the ends of DNA, and the endonucleases makes cut at a specific point within the DNA. What? Endonucleases acts within the DNA. Restriction endonucleases cut each of two DNA strands of double helix at a specific point in their sugar phosphate backbone. Each restriction enzyme recognizes specific palindromic nucleotide sequence in DNA. When restriction enzyme cut the strands of DNA away from the center of palindromic site, it produces overhanging stretches called sticky ends. What it's called? Sticky ends. The DNA fragments can be separated by a technique known as gel electrophoresis based on size. You can separate the DNA fragment based on their size by the process of gel electrophoresis. DNA fragments being negatively charged. What? DNA are negatively charged. Move towards anode. Anode being positive, DNA being negative. They attract both. Okay. Under electric field through the matrix in gel electrophoresis. The most common by used method is gel electrophoresis in agarose. Matrix is agarose, which is natural polymer extracted from seaweeds. From where we get agarose? It's seaweed. The separated DNA fragments in gel electrophoresis can be visualized after staining the DNA with compound ethidium bromide followed by exposure to UV rays. It is seen as bright orange color band. What? Ethidium bromide is used, UV rays are used, and a bright orange color band is formed. The process wherein the separated bands of DNA in gel electrophoresis are cut out from the agar jewel and extracted from the gel piece is called elution. The process of extraction is called elution. Origin of replication, that is ORI, is a sequence from where replication starts, and any piece of DNA when linked to this sequence can made to replicate within the host cell. From ORI, that is origin of replication, replication starts. Transformation is a procedure through which any piece of DNA is introduced into a host bacterium. Transformation helps to transfer the DNA into a particular host bacterium. When PBR322 vector is used for transforming recombinant DNA molecule using BAM-H1 as a restriction site, the recombinant cell will grow on ampicillin antibiotic containing medium but not on the medium containing tetracycline. They will grow on ampicillin but not on tetracycline. When what is used? PBR322 vector and BAM-H1 as restriction site. When a recombinant DNA is inserted within coding sequence of an enzyme beta galactosidase, it results in inactivation of the said enzyme, which is referred to as enjorsional inactivation. Okay. In case of unsuccessful enjorsional inactivation of coding sequence of beta galactosidase, the presence of chromogenic substrate gives blue colored colonies. When it's present, then it gives blue colored colonies. Okay, what blue colored colonies? Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA called as tDNA to transform normal plant into a tumor. Okay. Since DNA is hydrophilic molecule, it can't pass through the cell membranes. DNA is hydrophilic. Bacterial cells can be made to take recombinant DNA by incubating them with ice-cold solution of calcium chloride followed by heat shock at 42 degrees Celsius and then again putting them on ice. In microinjection method, recombinant DNA is directly injected into the nucleus of an animal cell. Microinjection is done for animal cells where it is put into the nucleus. In biolistics method, the plant cells are bombarded with high velocity microparticles of gold and tungsten coated with DNA. Gold and tungsten. Lysozyme enzyme can be used to break bacterial cells, cellulase to break plant cell, chitinase for fungal cells. Okay. PCR stands for polymerase chain reaction. Primers used in PCR are small chemically synthesized oligonucleotides that are complementary to the region of DNA. Primers. Oligonucleotides. The thermostable DNA polymerase used in PCR is isolated from bacteria's Thermus aquaticus. From where? Thermus aquaticus. If any protein coding gene is expressed in a heterologous host, what? Expressed in a heterologous host, it is called recombinant protein. Any protein expressed in heterologous host, it's called recombinant protein. If a continuous culture system, then the used medium is drained out from one side with while fresh medium is added from the other to maintain cells in a log or exponential phase. Log or exponential phase. Bioreactors are used for large volume, that is 100 to 1000 liters of culture and provide optimal condition for achieving desired products. The most commonly used bioreactors are stirred tank type. Stirred tank reactor facilitates even mixing and oxygen availability throughout the bioreactor. When you stir it, then the oxygen got mixed. In stirred reactor, sterile air bubbles increase area for oxygen transfer. The bioreactors have sampling ports so that small volumes of culture can be withdrawn periodically. After the biosynthesis stage of fermentation, further process includes separation and purification, which are collectively referred to as downstreaming processing. What's called downstream processing? The palindromic sequence of ECO R1 is 5GAATTC 3 dash prime 5 dash and 3 dash. That's a palindromic sequence. CTTAAG. Okay. So this is all about this chapter, biotechnology principle and process. I hope you like the session. Do like, subscribe to my channel. Thank you. All the best.